Good morning, friends. Good morning, students. In today's class, we will discuss mutations, right? We will also cover the types of mutation as well. So, first of all, uh, DNA replication is done with high fidelity, and DNA is a very stable molecule, right? But some agents that cause mutations are known as mutagens. Mutagens that cause mutations, and mutation refers to the change in DNA structure, right? And changes in this DNA structure can be seen in replication, transcription, and translation as well, right? These mutagens that cause mutations can be categorized into three categories. First, they can be physical agents, they can be chemical agents, or it can be also oxidative damage, right? So physical agents can be that cause mutations like UV rays and X-rays, right? And chemical agents can be some fungal toxins, right? And some alkylating agents, right? And oxidative damage can be induced by free radicals, right? Now we'll start with types of mutation. Now to categorize, there are two types of mutation. One is point mutation, and the second one is frame shift mutation, right? And the point mutation is further categorized into one is transition and the second is transversion, right? And frame shift mutation is categorized into two categories. First is insertion, A1, and the second is deletion, that is B1. Now to start with point mutation proper, point mutation is that you replace adenine, so you replace purine with purine or pyrimidine with pyrimidine, right? And there is single base alteration, right? So in transition, we have A and T here, adenine and thymine. Adenine is purine and thymine is pyrimidine, right? So here A is replaced with G and T is replaced with C, right? So adenine is replaced with G, so purine is replaced with purine, right? And thymine is pyrimidine and it is replaced with pyrimidine. So this is transition. In case of transversion, we have adenine and thymine here. So adenine is replaced with T. So adenine is a purine and thymine is a pyrimidine. So there is vice versa. Purine is replaced with pyrimidine. And in case T is thymine, pyrimidine, it is replaced with A, that is our purine. So in this case, transition purine can be replaced by purine and pyrimidine can be replaced by pyrimidine. And in this case, purine can be replaced by pyrimidine and pyrimidine can be replaced by purine, right? Now to start with frame shift mutation, uh, there are more than one type of base alteration, and for example we have A and T here, right? So this frame can be added or deleted further. So it can be categorized into insertion. Insertion means to add up a frame and deletion means to delete, right? So this frame is there, A and T, and there is further addition of T and A. So this is insertion, the category 1, and deletion, the whole frame is lost. So here A and T is not there, so this is our deletion, right? So that finishes with types of mutation. Now we'll start with consequences of mutations. Now we start with consequences of mutation, right? There can be three consequences. First is the mutation goes silent, and second is that it can go missense, right? And the third is nonsense, right? So first of all, we have code that is UCA, that code for serine, right? In this case, A is replaced with U, and and the amino acids remain the same because both code for amino acids serine, right? So this comes under silent mutation, right? Because the codon of messenger RNA containing chain base codes for the same amino acid, that is serine, again serine, right? And this is due to third base, right? UCA is replaced with U, right? So this is due to degeneracy of the genetic code, and there are no visible defects or problems seen in this mutation. So the name is silent. It doesn't cause any problem, so the name is silent, right? Now in second case, that is missense mutation, you have UCA here, that codes for serine. and in this case U is replaced with A, right? And ACA codes for the amino acid, that is 309, right? So in this case of missense mutation, the problem can be acceptable or partially acceptable, or it can be totally non-acceptable, right? How can you give example? The classical example is sickle cell anemia, right? And now coming on to third, that is nonsense mutation. In this case, you have UCA and the middle one C is replaced with A, right? So UA code is the termination codon, right? So it will stop the replication, and this comes under nonsense mutation, right? And the consequences of frame shift mutation are that there can be insertion or there can be deletion of the frame, right? So there is altered reading frame of messenger RNA is formed, right? So the messenger RNA does not know whether the base is missing or the new one, right? So because there are no punctuations in the reading of codons, right? So the proteins are synthesized with altered amino acids or it can be terminated. So these are the consequences of frame shift mutation, and that finishes our lecture. So please like the channel and subscribe it and share with your friends.